pharmacy in Mandsaur, we have done a lot of practicals of SOTEX, and we have seen research which is carried out by the Dr. Vivekanand Chattock sir. He is really a very dedicated researcher in the area of pharmaceutical science and research, and currently he is associated with the very good organization where he is doing a lot of research work, availing a lot of grant facility and all. So really congratulate you, sir, for your achievement. We are regular touch with you, and we are seeing your regular achievements, especially in pharmaceutical sciences. So I request Dr. Gurdeep Singh, sir, host of the today's scientific session, to kindly proceed with the introduction of Chattock sir.
Yes.
Please.
Very good morning.
Sir, sorry to interrupt you in between. Shall we run our video first? Is air on YouTube so that more and more participants also can join.
Okay, no problem. Please.
Thank you.
Very nice creation by Dr. Niharika Gokhale, ma'am. It's really so beautiful video
Thank you, sir.
of the International EFTP. So now I request Dr. Gurdeep Singh, sir. I think we should start our today's scientific session without any delay. Please, sir, go ahead.
Very good morning to one and all. On the behalf of Faculty of Pharmacy, Oriental University, Indore, I, Dr. Gurdeep Singh, extend a very sincere warm welcome to all the dignitaries present in the International Faculty Development Program. Today, we have very eminent speaker of pharmacy fraternity with us, Dr. Vivekanand Chattock sir. I feel privileged to extend my warm welcome to our first speaker of today's scientific session, Dr. Vivekanand Chattock sir. Dr. Vivekanand Chattock sir is working as an associate professor HR Patel Institute of Pharmaceutical Education and Research, Shirpur, Dhule, Maharashtra. Sir also served as ex-president APTI State Branch, Madhya Pradesh. Dr. Chatap sir has nearly 15 years of research and experience. He is a researcher with different vision. Dr. Chatap did his master degree from B.R. Nata College of Pharmacy, Mandsaur, and doctorate from H.R. Patel Institute of Pharmaceutical Education and Research, Sherpur. Sir has received grant of rupees 71.42, that is nearly about 272 lakh rupees from different funding agencies. He has guided more than 38 M-Pharma students and currently guiding 7 research scholars of PhD also. He has 4 patents and over 60 publications to his credit, which is published in reputed international and national journals. Sir is in editorial board members of reputed national and international journals. Sir has received so many awards like Best Innovation Awards in 2017, APTI State Branch Award in 2015, Young Researcher Award, Young Scientist Award, and so many. Sir has published textbook of pharmaceutical biotechnology, and his major field of scientific interest are solubility enhancement, nanocomposite, nanofibers, and biomaterials for drug delivery system. So there is a lot of talk about the Vivekanand Chatap sir, but without delaying the session, I would like to call our dynamic Dean in charge, Dr. Nitesh Jain sir, Faculty of Pharmacy, Oriental University, to welcome Dr. Vivekanand Chatap sir. Over to you, sir.
Thank you, Dr. Gurdeep. So once again, on the behalf of Faculty of Pharmacy, Oriental University, Indore, I heartily welcome Dr. Vivekanand Chatap sir, our mentor, our senior, our well-wisher. So, sir, I'm really very grateful and thankful to you for accepting our invitation as keynote speaker in International Faculty Development Program. Hope your title is very good, sir. Sir is giving presentation on role of phytochemicals, phytoconstituents, especially in the drug development. So I hope every researchers will get some ideas about the future research and all. So thank you so much, sir, for accepting our invitation. And we are lucky to have, and we will listen you today as a keynote speaker. Thank you so much, sir.
Thank you, Dr. Nitesh Kumar Jain, sir. First of all, I would like to thank you and your team for organizing this international faculty development programs in this pandemic situations. And very good, you have selected a good topic, that is integrated approach to natural resources in drug development and drug discovery. It is the current need of our pharma people and researchers. As we know, most of the NDA, abbreviated new drug delivery drug, are expired or patent are expired, and now the research is currently in, that is the international as well as in domestic market, the whole drugs are already exhausted. Okay. That means, and the new drugs is little bit, it comes from the synthetic source. So, and for development of single drugs, it's need the 7,000 crore rupees. So it is no, and also time should required also. And currently, most of the drugs, those are available in market, is divert the drug discovery and drug development, that is formulation development parts, towards the from tablet, solid, and semi towards the that is injectable dosage forms. That's why this demands is increasing into the injectable current situation, injectable formulations dosage form. But as a point of view in natural resources, that is the good examples. Today we are going to discuss with you peoples to, that is the, which are the sources, that is the main source of any drug nowadays, that is the natural resources. As we know, that is the cancer, diabetes is a chronic diseases. But the 60% of the drug which are currently used in treatment of this chronic disease, that is the cancer and diabetes, is from the natural origins. That is the current situations and demand for the treatment of this such current diseases. And for that purpose, I would like to thank Dr. Nitesh Kumar Jain sir for inviting me as a resource person.
So
And this,
it's our honor. Matlab,
that is,
we are lucky to have you today as a keynote speaker.
yeah. So for this international faculty development program, and I would like to thanks Dr. Kuldeep Singh sir for that is introduced me very nicely. Uh, Niharika madam, Sumit sir, Apurva madam, and Dubey, Anand Bele sirs, uh, and my dear students. Today I am going to deliver the topic entitled, that is phytochemicals in new drug development. Can I start the share the PPT? Yes. Okay. Today I am going to deliver the topic entitled, that is phytochemicals for new drug development. Uh, in this today's lectures, today's that is interactions, we are going to talk about the sources of phytochemicals, then types of chemicals, then what are the challenges in phytochemicals, and new advancement in phytochemicals with respect to the new drug development and new drug discovery. Now, as we know, the plant source is the main source for natural drugs for the or natural resources, and plant remain the most important source of the natural drugs which is available in the markets. However, only 10% of drugs is fully studied yet. That means we have the lot of opportunity to do the research in this area of natural resources, not only in plant but have the several resources, that is the plant, that is animal origin, marine origins, several origins we have natural resources with us. So that is the lot of chance to explore them and do the research and make our futures bright in our pharma professions. And more than 30% of prescription is nowadays the drugs is obtained from the natural products. As we know, so many small companies or entrepreneurships or drug entrepreneurs are comes into the market, and most of them are herbal origins products. They are not the prescription also, but they are selling directly. Okay, in that is PCT markets or PMT markets is most of the things. Okay, and as we know, that is Patanjali Ramdev Baba is a well-known, that is the now market leaders. Here, that is, he has crosses the 5,000 crore rupees turnover company and expected to 10,000 crore rupees, that is turnover is going on. That is the, it's based on to the herbal origins or the natural resources. He is work fantastically from 5 to 10 years and explore the only naturally that is natural plants, okay, and minerals, etc. That is the natural resources they are explored for the drug discovery and the drug development purpose. So our researchers have a lot of opportunity to explore in that the area. And more than 60% of drugs, that is, as we discussed in earlier, that is anti-cancer, anti-infective, anti-diabetics, we are totally dependent on to the natural products, not for the allopathics. Because of once we have to go for the allopathic treatment for this chronic diseases, that is as like as a cancer or anti-diabetes, we have the that is disadvantage also, may chances of toxicity, as well as that is harmful effects on our body. And these allopathic treatment are costly as compared to the anti-diabetic, as compared to the natural resources or the herbal or plant origins is there. So therefore, the more than 60% of drugs still are used for these chronic diseases for the natural products. As we know, that is main sources of drugs which obtained from the natural sources are, that is three, that is natural sources, semi-synthetic sources, synthetic sources. An example of natural sources which drugs include, that is plant origins, microorganisms, marine plants, animals, obtained from the natural source. Any others we can be that. Then semi-synthetic substance, which is the combination of the natural products and the synthetic molecules. Once the scaffold is considered as a natural source, you have to link together or join together and make up one compounds, synthetic compounds, which is available in market, that is the steroidal hormones and corticosteroids, and this have the good market in domestic as well as in international markets. And synthetic drugs which is obtained from the totally, that is the from synthetic origins, as well as called as that is the from total synthesis. The phytochemicals, what do you mean by the phytochemicals? Phytochemicals are the chemicals which is obtained from the natural sources or that is specifically in plant origins. They are chemically or naturally present into the plants, and itself they acts as a protective compounds that may help to prevent the disease. Phytochemicals are present into the plant in very small quantity, but which but it is very potent, which is present into the plant, which may act as a, that is drug itself or the immunizers, you can say that, okay, which increase the immunity of the plant and protect from the any other environmental disease which may be affected to the plant, as like as the drug effect that can influence our body chemistry. In example, you can say that that is the plant, some of the plants are bitter in nature. Okay, that means these plant contain alkaloids. And why they are bitters? If any that is insects are sit onto the plant to eat that plant, the plant herbs, that is the bitter in nature, so they can't bite or they can't eat that plant. So itself that is protect the plant because of increasing the concentration of the alkaloids. That is the protective mechanisms of this, which it helps various environmental factors. As like as a drug, if any that is our body conditions is that is disturbed, okay, that is blood sugar level increase or decrease, or either that is the affected from any gastroenteritis problems, that means our system has been disturbed. In such conditions, we are taking the drugs and normalize our systems. That's why we can say that that is the alkaloids or glycosides. All these are present. Amino acids, lipids are present into the plants, which is acts as that is some of the compounds which is acts as a growth of development of the plant. Some of the compounds which is as a preventive of the diseases. The plant mainly consists of two types of phytochemicals. There is primary phytochemicals, or you can say that primary chemicals or primary metabolites, and secondary chemicals or secondary metabolites. In primary metabolites or primary chemicals, compounds which can be used or which can be helps to improve the growth and development of the plant is there. Okay. And in secondary chemicals or the secondary metabolites, these are indirect helps to development of the plant, but they are mostly helps to protect the plant or prevent the plant from the any diseases or disorders. Anyone knows that is the primary chemicals, examples of primary chemicals? Okay, I will go further. That is the primary chemicals. Primary chemicals are the molecules that are essential for the growth and development of the plant. and which are necessary for the basic survival of the plant and used for the energy and the tissue constructions. Here the word is used as a tissue construction. As we know, all these things, all these words are very, very well known. But as tissue construction, some of the chemical compounds are present into the, there is plants which can be used for the regeneration of the cells also. Okay, and that compounds which can be used for the in our drug delivery or drug development for it will be helps to regeneration of the cells. Example, best example as we know from the natural sources, that is gelatin. Gelatin have the property to regeneration of the cell, regeneration of the cells. Okay, therefore the gelatin is used in the as a biomaterial for the drug delivery or reconstruction of our human cell is there. Okay. These are the different examples of the primary chemicals or primary metabolites, which includes that is most of the carbohydrates, amino acids, protein, lipids, nucleic acids, and some vitamins. As we know, these are the primary compounds should required for us also for development of our growth and develop for growth and our essential growth and our human body developments. Okay. Similarly, as a plant also. And now that is the secondary metabolites. Secondary metabolites
आपने कलर और इसके दो की फोटो दिए ना, बाकी उसकी फोटो बाकी मेरा नहीं, अच्छा नहीं तो आपने उसमें प्रतीक और ये उसका भी तो दिया हुआ है।
प्रतीक आपकी आवाज आ रही है
इसमें और कुछ आया हुआ है वो उसने ऐड कर दिया वो मैंने रोल ये सेकंड सेम में मेरा एक बच्चा है बिलाल का, नेक्स्ट सिर्फ बिलाल उसका मैंने आपको करण का दिया आपने। हाँ। इनके तीनों के वो दे मेरे बच्चे की दीपिका के और दीपिका ने बना के दे दिया ना उसी में है ना।
Thank you.
I'm really sorry, sir.
Okay. Thank you. No problem. Secondary chemicals or secondary metabolites is mostly obtained from the pieces of the primary metabolites. It is small concentration which is be obtained or that is synthesized during the metabolism process of primary metabolites, and this is compounds or the potent compounds which will be helps to prevent the disease of the plant is there from various environmental factors, and they are more prevalent or the unique or the certain genus or the species or the families which is present in the each plants. The some compounds are present in particular plant, that is single compounds are present in particular single compounds, not into any other. Okay, likewise. Then indirectly involves in plant growth and the developments, which is the helps. That means if it is used as a protective, if any disease are infected onto the plant, that means growth is reduced. But in the same time, these phytochemical secondary metabolites, it's prevent from the various prevent the plant from the disease. That means there is not decrease the growth of the plant while increasing the growth of the plants. That is the indirectly helps to increase the growth of the plant is there. These are the diverse group of the molecules that are involved in adaptation of the plants and their environments, involved in defense against herbovirus and the pathogens. Though some of the plant virus and plant pathogens are affected onto the plant and it's become the disease conditions or into the plant. It's the secondary metabolites or secondary chemicals, they are protect themselves. Okay. As like and same this chemical secondary metabolites are chemical components which is used to protect from the plants. Likewise, these if it is these glycoside alkaloids which can be used for the treatment for the human beings, they can also protect us. Therefore, most of the plant origin compounds can be used for the treatment of our disease conditions in human beings and recovery of regeneration of our body also. The examples of secondary chemicals, that is glycosides, flavonoids, alkaloids, tannins, terpenoids, steroids, and so many is there. Phytochemicals for the new drug development. That is, what is the applications part? This is up to that. That is the part of in today's lecture. That is the today's presentation is basics. Now this is the advancement. What are the advancement and application part of natural resources? How the natural resources can be used, or phytochemicals, primary or secondary phytochemicals can be used, and applications into the drug discovery and drug development is there. And natural products is remain the important source of new structures, though not the final drug entity. As we know, so many chemical active compounds or activity we can be carried out, animal activity, and that is the in vitro activity we have to carry out, and we have tried to prove that that chemical component is become the effective, more effective against the particular any disease conditions or the viruses or the bacteria, microorganisms itself. And we have proven that this the phytochemicals or the chemical constituents present into the natural products, natural resources may more effective into that particular pathogens against the pathogens. But this is not the final compounds you called as. Okay, in such compounds, that is so many molecules or the compounds are present. So in such condition, need to isolate them. Then that isolated compound, example that is vincristine, vinblastine, everybody is the famous alkaloidal, and which is used in treatment of the cancer is there. Okay, we are learning from the V form. Okay, same things. So that is the isolated compounds which are obtained from the plant vinca is there. Similarly, that is the need of that compounds, but these compounds are the need to improve the efficacy in such conditions, need to development of the or synthesis of the semi-synthetic versions need to develop is there. Okay, example we can show in next slide, that is the scaffold. We can consider as this chemical compound is considered as a scaffold for the natural compounds, that is secondary metabolites, and serve as a guide for the development of different derivative of the molecules. यानी एक अगर सेकेंडरी मेटाबोलाइट्स का कंपाउंड से वो आप स्कैफोल्ड कंसीडर कर सकते हो, उससे फिर आप बहुत सारी अपॉर्चुनिटीज है उसमें ग्रुप मॉलिक्यूल कर सकते हो, फिर उसमें डेवलपमेंट हो सकता है और डिफरेंट डेरिवेटिव्स प्रोडक्ट्स डेवलप कर सकते हैं. As we know, that is so many persons, if you know, aware about the docking systems. Okay. Similarly, these docking systems which are developed for the total synthesis or the synthetic origin molecules, but the docking, molecular docking, we can be applied for this scaffold which is obtained from the natural origins. Okay. And we have synthesized so many molecules in molecular dockings. In that, which one proteins molecule is that is shows the affinity, proper affinity, or the lock and key system is block, and which will shows the proper affinity, effective efficacy in such products can be synthesized. Then after synthesize, we can go for that is semi-synthetic product can be derivatives developed from the natural source. Then we go for the activity. Then we go for the development of the product development. Semi-synthetics of the parent molecules solve the problem of targeting as well as toxicity. If the natural origin compounds have the problem related to the toxicity as well as that is targeting problems, you can be solve the problem of targeting to the specific affinity and specific bindings as well as reduce the toxicity by molecular dockings with the scaffolds. This is the examples we can see, that is the here this is the scaffold, consider as the scaffold of the phytochemicals, and after that we can develop the so many derivatives. That is, as we know, these are the well-known drug which is semi-synthetic drugs which is obtained from the plant origins and available into the market. That is lovastatin, simvastatin, pravastatin, fluvastatin, that is cerevastatin, atorvastatin, rosuvastatin, pitavastatin. These are the lipid lowering compounds which can be, that is booming the market into the, that is domestic as well as international markets into the in current situation is there. And some of the important secondary metabolites serve as a milestone in phytochemistry. That is morphine, as we know, morphine, that is strychnine, quinine. These are the, that is the compounds which are more research is already that is explored. But as I told in first slide, till date only 10% of the drug is explored into the plant origin. We have the opportunity to explore 90% of the resources, natural resources, and to the research. Okay. Then important secondary metabolites in another, that is the cholesterol as well as that is ephedrine. As we know, that is the cholesterol, that is the cholesterols which is obtained, that is the steroids which is obtained from the gallstones. Then we have to convert it into the, that is 7-dehydrocholesterol, that is provitamin D, and that is finally that is vitamin D3. And vitamin D3 for administration. Then after that we will metabolize in our body. Then we receive the body, our body is received the vitamin D is there. Okay. Then what are the challenges with us in the natural products today? That is the major, these are the three are the major challenges in front of us, and it's need to work on that. Okay. So for the development of drug discovery and drug development from the natural origins, these three are the challenges in front of us. First is that is loss of biodiversity. In loss of biodiversity means that is some of the species are more, that is totally explored. Okay, so that's why the loss of this origin. So it's need to protect that origin. That is new plantation of the drugs, particular species you need to cultivation and development. That is the need. Otherwise, that specific plant is, that is the totally removed from the earth is there. So that is cutting off the plants, that is affects the loss of, that is totally the biodiversity is losses there. And especially that is improvement in quality. That is another challenge in front of us. So many products are available in market, that is Ramdev Baba products, and so many herbal products and natural based products are available in market. But the standardization is a major issue current till date. This is the challenge in front of us. And the standardization procedures as per the WHO and FDA, it is not specified though, and in that standardization have some loopholes. Example, that is we can say that that is the most of the now that is recently I have the seen the that is crisis in the Dabur. Okay, Dabur and good companies nowadays, that is the Dabur honey have mixed alternative products. Okay, that is the situations. It need to provide the purity, how to standardize the purity of any natural products. That is the issue. And some the industry or good industry have find out the loopholes in that and take the benefit and comes into the market. They are selling the products into the market, but have some, that is the standardization problems, or you can say that that is most of the natural products which is available in market, how the concentrations is more. Okay, but showing is less, but it is present. It's how that is the more concentration are present. That is the need to improve the quality of the products. For that purpose, specifically that is standardization process you need to develop itself. Another, that is elucidation of mechanism of actions. Some well-known compound have elucidate the mechanism of action, but you can say that 90% of the natural drugs is available into the market. They are not elucidate the mechanism of action of the natural product, how they work. Once administration into our body, how they are metabolized, how they are absorbed the drug, this herbal drug or phytochemicals, primary metabolite, secondary metabolite is sold by the body. This mechanism is doesn't know that. Okay, it's not that is proven. This particular mechanism action of the drug, particular natural resources of drug is obtained from natural resources, and mechanism action of this particular drug is there. It's haven't elucidated. It's the that is in front of us have the challenges to that is the discovered it. Okay, as we know that is the we have read the books, so many books of medicinal chemistry, and as well as the pharmacology is mentioned that the pharmacological action of this is not yet known. Okay, likewise not discovered. Similarly, conditions of this natural resources products is not discover or more explore the mechanism of action. It's our that is focus area. We can focus on that and discover that. And this is the last slide of today's presentations. That is the recent advances, or we can say that the research area for the natural products in today's scenario. We have the lot of opportunity for the researchers to work into the natural resources or natural origin product. That is like a structure elucidations. Okay. Once we have to, so many compounds has been proven for the particular activity is carried out, and we can say that is become the that are used for the particular treatment is there, but it is not particularly isolated the single compounds and elucidate. It's need to elucidate the structural compounds, that is need to determine mass, NMR, FTIR, LCMS, and this is the requirement for analysis of the drugs. Nowadays so many advancement techniques are available, LCMS, MS, okay, and mass. So many techniques are available for the structure of the natural compounds. So use that techniques and elucidations should be required for this today's scenario for the natural products. Another, that is the metabolomics. Metabolomics is the large scale study of small molecules, which is commonly called as that is the metabolites. That is nothing but the metabolomics. And we call it within the cells, biofluids, and that is tissues of organisms. These are includes into the metabolomics. And collectively, the small molecules and their interactions within the biological systems are known as metabolomes. Okay, so need to work on this area also. These small molecules, how it's works after administration of the body, how it's works, how it's get reacted or with our enzymes present in our body, which produces the therapeutic efficacy. Likewise, then that is the biological activity. As we know, that is the need to work. Once you have to prove that that is the theoretically it is affected, so reported in pressures, it is good biological activity, but it's need to prove that again itself in animal activity during preclinical study, then in clinical study. That is that study it's need to focus with respect to the natural products. As example, so many that is natural resources which can be used for that is the cancer, as well as that is the diabetes. But so many Ayurvedic persons are given the medicines, or Ayurvedic medical stores are that is dispensing their we call it as Ayurvedic medicines for the treatment of diabetes and cancers. The patients are recovered from this natural origin base, that is dispense the medicaments or the natural origin products from the dispensary or the medical shop, Ayurvedic medical shop, but they doesn't know that how they act it. So need to prove that the biological activity of that compound. Another is molecular biology and biotechnology. We have the good opportunity. Specifically, that is the work on to the Department of Biotechnology is currently working on to the that resources, that area of natural resources. They are going to explore the how can we biotechnology is involved into the plant-based biotechnology, which can be that is explore to discover the new that is molecules from the natural source origins and use for this drug development. Okay, and the molecular biology, that is the bioanalytical development area, is nowadays is that is booming area. We have the lot of opportunity to work in that area. So this is the good research area nowadays, molecular biology as well as that is the biotechnology. And quantitative natural product chemistry, if we go for the as we know this till date, the chemistry of natural products is less explored, or we are not, once we have to go for the research in that is M.Pharm and the PhD, but we are not good in details. Okay, so it's need to specifically work on to the natural product chemistry. We have the lots of opportunity to work into that research area, and hope which will be expected more into that, find out the good things into that area. Okay, and that is the finally our nature is the supramolecular architectures or architects. So we have the lot of opportunity to create the research opportunity into the natural resources and make the futures into the bright. Thank you. If anyone have the questions, we can ask. Welcome.
Hello, sir. Sumit here.
Yeah, बोलिए सर.
सर मेरा एक बहुत सिंपल क्वेश्चन बेसिकली यहाँ पे बहुत सारे रिसर्च स्कॉलर्स हैं उनके लिए था कि बेसिकली बीइंग इन फार्मेसी वी जस्ट गो विद द biological or pharmacological screening of the plant or the plant extract. Why we are not going for the formulation aspects of the same? So in this view, will you light something? मीन्स नॉर्मली हम लोग क्या करते हैं कि प्लांट के एक्सट्रैक्ट की फार्माकोलॉजिकल एक्टिविटी करके छोड़ देते हैं, कोई सा भी रिसर्च स्कॉलर हो, बट फार्मुलेशन एस्पेक्ट्स में वो नहीं जाता है. तो सर मीन्स उसमें क्या कुछ, बीइंग सिंस यू आर एक्सपर्टाइज इन फार्मास्यूटिक्स, दैट्स व्हाई आई एम आस्किंग कि क्या उसमें कुछ बेसिक दिक्कतें आती हैं हर्बल्स को लेके खास तौर से, कि जिसके कारण कि रिसर्च स्कॉलर्स भी फार्मुलेशन एस्पेक्ट्स में नहीं जाते हैं, और क्या उनको जाना चाहिए उस चीज पे?
It's a good question. This question is with respect to my area of interest. सर मैं हिंदी में आपको बताना चाहूंगा कि जो आप यहाँ तक के जो फार्माकोजेनिक एक्टिविटी सब हो जाती है ना, तो उसके बाद में जो फॉर्मुलेशन डेवलपमेंट आता है उसमें सबसे बड़ी दिक्कत ये होती है, जो भी आप प्रोडक्ट बाहर निकालते हो उसकी प्यूरिटी का प्रॉब्लम आता है. उसमें इम्प्यूरिटीज और इनर्ट मटेरियल बहुत सारे होते हैं. अब अगर अपने अगर ये सोचे कि भाई कोई अगर एक पार्टिकल, कोई एक अगर लीफ लिया, उसका अगर क्रश किया, उसको एज एक्सिपिएंट करके यूज कर, लेकिन उसमें बहुत सारी चीजें ऐसी होती है, इनर्ट मटेरियल होते हैं, उसकी इफिशिएंसी जो है वो कम कर देते हैं. तो कहने का मतलब ये है, अगर ड्रग अगर आप अगर फार्माकोलॉजिकल स्क्रीनिंग करते हो, वो स्क्रीनिंग जो आप करते हो उसमें और डेवलपमेंट की जरूरत होती है. तो कि एक जो आप एक स्पेसिफिकली ग्लाइकोसाइड या फ्लेवर, एक प्लांट में ग्लाइकोसाइड भी हो सकता है, अल्कोलाइड्स भी हो सकता है, फ्लेवोनाइड्स होते ही है, एंटीऑक्सीडेंट भी होते हैं. यानी एक प्लांट और एक प्लांट का कोई एक पार्ट है, सपोज यानी लीफ है या बार्क है, उसमें ये सभी चीजें हो सकती है. आप अगर स्क्रीनिंग करते हो आपको स्टडी मिलेगी, इतना ये कॉन्सेंट्रेशन है, फ्लेवोनाइड्स इतना है, ग्लाइकोसाइड्स इतना है, एंटीऑक्सीडेंट इतना है, बहुत सारी चीजें होती है, छोटे छोटे कॉन्सेंट्रेशंस में होते हैं. मैंने पहले लेक्चर्स में बताया कि secondary metabolites are the present into the plants is very in few concentration, but they are very potent. in such conditions need to isolate the single compounds.
बट
आप
सर
अगर
सॉरी
उसको
सर
अगर
सॉरी टू
अगर कर
इंटरप्ट यू।
सकते हो।
जी जी
Yes,
सर, मैं थोड़ा सा यहाँ पे रोकना चाहूंगा। मींस जैसे अपन
हाँ।
हर्बल फॉर्मुलेशंस की बात कर रहे हैं, ठीक है ना, बट व्हेन वी आर आइसोलेटिंग एनी कंपाउंड फ्रॉम द प्लांट, इट शुड इट बी नो लॉन्गर हर्बल, ओके। क्योंकि जो WHO गाइडलाइंस में हर्बल की डेफिनेशन दी है वो ये दी है कि एज सच आप प्लांट के किसी भी एक्सट्रैक्ट को या पार्ट को यूज करो, देन ओनली इट बी हर्बल। अगर आप आइसोलेशन में जा रहे हो, है ना, तो वो हर्बल नहीं हो जाता। मैं तो सिर्फ आप सर, इसलिए मेरा मेजर कंसर्न हर्बल फॉर्मुलेशन के ऊपर है। आइसोलेट करके नो डाउट वी कैन गो फॉर वेरी गुड फॉर्मुलेशंस।
मैं यही चाहता हूँ, अगर उसकी अगर इफिशियंसी अगर आपको अगर ज्यादा चाहिए प्रोडक्ट में, अगर उसको अगर फॉर्मुलेशंस में आना है तो प्यूरिटी का इश्यू आता है। अगर हाईली प्योर्ड कंपाउंड रहेगा, आप अगर उसमें फ्लैश क्रोमेटोग्राफी यूज करो, डायरेक्टली स्क्रीन आता है, उसमें आप सिलेक्ट करते हो, सिंपल में एक ग्राफ आता है, जो वो पीक में आप सिलेक्ट कर लो तो कंपाउंड डायरेक्टली सेपरेट हो जाते हैं। वो कंपाउंड को आप आगे और स्टडी परफॉर्म करो, जो कंबाइंडली आपके पास में जो एक्सट्रैक्ट आया था उसकी बायोलॉजिकल एक्टिविटी अलग आएगी, और अगर आपको अगर यानी आप अगर सिंगल करोगे तो उसकी एक्टिविटी आपको अलग आएगी। तो ये जो डिफरेंस है, फिर उसको आप फॉर्मुलेशन डेवलपमेंट में चले जाओ तो उसकी जो इफेक्टिविटी और इफिकेसी है, थेरापेटिक इफेक्ट्स है, वो कांस्टेंट रहेगा। वो क्योंकि जो डेवलप्ड एक्सिपिएंट है, जो डोमेस्टिक मार्केट और अभी अपन जो डिफरेंट फॉर्मुलेशंस में यूज करते हैं या इंटरनेशनल कोई भी मार्केट में अपन जो यूज करते हैं, उसका केमिकल इनर्ट और दे आर प्रूवन एक्सिपिएंट्स, उसमें आप थेरापेटिक इफेक्ट में या कोई अफेक्ट नहीं करते। तो अपने को जो नेचुरल रिसोर्सेज जो प्रोडक्ट मिला है उसी के साथ में आपको कंबाइन करते हैं और फॉर्मुलेशंस इजी हो जाता है। आपका जो उसके साथ में आप प्री फार्मुलेशन कंप्लीट कीजिए। प्री फार्मुलेशन स्टडी कंप्लीट करने के बाद में ऑन द बेसिस ऑफ प्री फार्मुलेशन स्टडी we can choose that or we can select that which formulations we are going to prepare. That is, we are going to for prepare tablet, emulsion, suspension, that is anyone those you want to prepare it, but based on to the that is physicochemical properties of the drug we can be select. On that basis, which dosage form you are going to select. On that basis, first we have to select the dosage. On that basis, we have to select the available excipient and make a formulations. Then you have to go for the again activity, whether it is similar to the previous one activity of that compound or not. That is the things is there. Yeah.
सर एक लास्ट क्वेश्चन मेरा और है।
So this is
जी सर, एक
area and need to do research opportunity in that.
ओके सर, एक लास्ट क्वेश्चन मेरा और है।
हाँ सर बोलिए।
जैसे सर इन केस, चलिए अपन ने अगर कंपाउंड को सेपरेट कर लिया, आइसोलेट कर लिया तो वेल एंड गुड, नो डाउट एट मींस सर्टेन एक्सटेंट इट इज गिविंग वेरी गुड एक्टिविटी। डिस्पाइट ऑफ दैट, अगर हम एक्सट्रैक्ट को लेके फॉर्मुलेट करते हैं, ठीक है ना, तो उस कंडीशन में उसका डीजो कैसे चेक करें? मतलब मेरे साथ पर्सनली बहुत बार इश्यूज आए हैं कि अगर, क्योंकि एक्सट्रैक्ट को लेके अपन फॉर्मुलेशन तो नो डाउट है बना रहे हैं, बट व्हेन वी आर गोइंग फॉर द ड्रग रिलीज, तो किस ऑप्टिमम वेवलेंथ पे अपन उसको चेक करें, वो एक थोड़ा सा दिक्कत आता है। तो उसके लिए अपन सर क्या कर सकते हैं?
सर सिंपल सा फंडा है, यूवी जो वर्क करता है वो कलर बेसिस पे, आपको पता है ना। व्हाइट तो कलर वेरिएशन विजिबल लेंथ रहती है, जो कलर कंपाउंड से उस बेस पे आपको यूवी की वॉल्यूम यानी वैल्यू देती है, अपने को वेवलेंथ देती है, बराबर। तो पहले आपको स्टैंडर्डाइजेशन करना पड़ेगा, कैलिब्रेशन करना पड़ेगा, जो ड्रग आपको एक्सट्रैक्ट जो है वो, ठीक है। पहले आपको यूवी एनालिसिस करना पड़ेगा, उसकी वेवलेंथ डेवलप करनी पड़ेगी, फिर वो सेम एक्सट्रैक्ट आप जब यूज करते हो फॉर्मुलेशंस में उसका डीजो करो। जो पहले एक स्टैंडर्डाइज आपको कैलिब्रेशन कर्व डेवलप करो, डिफरेंट कॉन्सेप्ट, फिर आप वही स्टैंडर्ड कैलिब्रेशन कर्व आप यूज कर सकते हो उसी फॉर्मुलेशंस को डीजो करते वक्त। और प्रॉब्लम्स क्या होता है, आप मैं भूल गया आपको कि ये प्रॉब्लम्स क्या होता है, यानी हर्बल फॉर्मुलेशंस में, इसमें रिप्रोड्यूसिबिलिटी ऑफ वर्क नहीं है। बड़ा पैच है, आपने एक बैच लगा दी तो वो बैच, वही क्वालिटी के बैच, वही इफेक्टिव बैच दोबारा बनेगी, इसकी अपन गारंटी नहीं ले सकते। ये दूसरा ऐसा है जो एक्सट्रैक्ट द कंसंट्रेशन क्या है, डिपेंड्स अपॉन द डाइल्यूशन है। होम्योपैथी का कंसंट्रेशन, होम्योपैथी का कंसेप्ट अलग है, आप जितना डाइल्यूट करेंगे उतना कंसंट्रेशन उसका बढ़ता है, लेकिन आयुर्वेदिक में जितना डाइल्यूट करेंगे उतना कंसंट्रेशन कम होता है, की तरफ जाता है, ये डाइल्यूशन की तरफ आता है ना। तो इसकी वजह से सबसे बड़ा दिक्कत तो यहाँ ही है। तो आप क्वांटिटी जो भी एक्सट्रैक्ट ले रहे हो उसमें आप क्वांटिटी मेजर करना जरूरी है, उसमें कितना कंसंट्रेशन, कितना mg माइक्रो mg ड्रग है, तभी आपको ये पता होना चाहिए कि जो डीजो ले रहे हो उसमें आपने कितना कंसंट्रेशन डाला है। 50 mg कंसंट्रेशन डाला है, आप जब सैंपल विड्रॉ करोगे तो आप स्टैंडर्ड कल निकला है उसमें से वैल्यू आ जाएगी कि इतने मिनट में आपको इतना कंसंट्रेशन का ड्रग मिला है। लेकिन एक्सट्रैक्ट में क्या होता है, अपने को एक्सट्रैक्ट अभी पहले पता नहीं रहता है कि
बिल्कुल जी सर,
कंसंट्रेशन ड्रग है उसमें। तो
actually sir, various chemical constituents are present in extract, so it is very much difficult to do the analysis on certain particular wavelengths, sir.
Yeah, that is the problem in
herbal formulations. Yes sir,
that's why I'm saying that first you go for the isolated compounds.
Yes,
Single compound it's better, but it is if it is, would you like to give the any research topic to any your research scholar, so one research scholar should be work up to that part, that is biological, that is phytochemical study. One should be study for the phytochemical screening only, one should be study for isolation, and one should be work for the formulation development. That should be divided. But इसके लिए आपको क्या करना पड़ेगा, अभी एक साथ में नहीं दे सकते हो। अभी एक स्टूडेंट ने आपको अगर फाइटोकेमिकल स्टडी स्क्रीनिंग तक का अगर वर्क उसने कंप्लीट कर लिया है तो उसको आप आगे का काम दो। अगर दूसरे स्टूडेंट ने वो काम कर लिया है तो वो उसको आगे का काम दो। आप एक ही स्टूडेंट को पूरा डिवाइड करके नहीं दे सकते,
नहीं दे पाएंगे सर।
उसको वो पहले डाटा मिलेगा, वो डाटा और उसके ऊपर आगे का काम करेगा, तो उसको लिए ये ज्यादा बेनिफिट होगा।
डेफिनेटली सर। ओके सर, थैंक यू सर, थैंक यू।
ओके, इफ एनी क्वेश्चंस वी कैन आस्क me for telephonics or email with us, no issue about that.
Definitely sir, anytime sir.
Yes sir. Thank you
in future sir.
The screen वैसे तो इंदौर में आते रहते तो मिलते रहते, लेकिन अभी स्क्रीन पे सेशन हो गया है और अभी मिले ऐसी लग रहा है।
डेफिनेटली जब भी नॉर्मल कंडीशन होगी आप आएंगे, हम बिल्कुल मिलेंगे सर आपसे इंदौर में।
बिल्कुल मिलेंगे, चलो।
Gurdeep sir.
Yes ma'am. So once again I thank you sir for such a valuable and knowledgeable session regarding the implementation of natural resources, especially with the formulations. Now I request Dr. Nitish Kumar Jain sir kindly felicitate Dr. Vivekanand Chatap sir as a token of respect sir. Please sir.
So thank you Dr. Gurdeep Singh sir, and again it's an honor to give some kind of appreciation or the acknowledgement to our beloved mentor seniors, Dr. Vivekanand Chatap sir. So sir, on behalf of Faculty of Pharmacy, Oriental University, Indore, so this is a kind of token of appreciation and our actually just affection towards your personality, and definitely today scientific session was really very excellent, and you have given a lot of tips for the new innovation based on the natural resources, especially in the drug discovery and development. So thank you so much sir once again accepting our invitation as a keynote speaker, and you have given valuable insights on the topic that phytoconstituents which basically used in the drug discovery and development. So thank you so much sir. Thanks to you sir.
Thank you sir, thank you for your attention. Thank you, welcome.
So here we wrap up our today's first scientific session, and the next session will start at 3:30
Can I leave sir?
Sorry sir.
Yes sir.
Can I leave sir?
Yes sir, thank you so much sir. Thank you. Yes.
Okay sir,
Thank you sir.
Okay sir, thank you.
Thank you everyone. to join with us, and we will meet at 3:30 p.m. today with the next today's second scientific session of Sally Al Jahabi. Thank you so much.